Thank you for inviting me to give a lecture here in the Mendel series. This is a distinct honor. It's a great pleasure to be here at this place. Maybe before talking about some serious science here, let me begin with a little anecdote. As already mentioned, I began to study as an undergraduate at the University of Muenster in '66. Now, at that time, the University of Muenster did not have any curriculum for biology. And the good thing was, in retrospect, I think this was much better than, what happened at other places. That physicists, chemists, and biologists took all the same courses for the first 2 years of their study. Then, nevertheless, for some reason, I moved to the University of Bonn, which, in some respects was maybe a mistake. They had a curriculum in biology, but this curriculum was awful. We were sitting from morning to evening looking through light microscopes and trying to draw, plant tissue, plant cells, whatever we saw there. And I think the performance of the students was, evaluated by their ability to sort of give the right shading. So it was the beauty of the drawings we made, and I have to admit that in that respect I was an underachiever. So I was looking for something a bit more interesting, and I think that brought me to this person here on the right-hand side. His name was Franz Weiling. He was a professor in the Department of Agricultural Botany at the University of Bonn. He had a couple of, common background, if you like, with Mendel, in the sense that he originally had studied theology or divinity, but at the end of the day, he did not become a priest for very human reasons. And so he studied then, mathematics, and he studied botany. And so he became what, it was called biometry at the time, or statistical biology, or whatever. So he gave a lecture series which was sort of very demanding. It was usually, he had, at the beginning of the semester, he had maybe 4 students. At the end of the semester, he had usually 1 or 2 students who sort of survived the course. And I think that sort of introduced me to, Mendel, because it was the time when there was still the famous Mendel-Fischer controversy. It was about, the question whether or not, the data, the experimental data of Mendel were just too good, okay, so to be true. And many dealt with that, but Franz Weiling made it the mission of his life to use his skills in mathematics to sort of re-evaluate, Mendel's experiments. And he came to the conclusion he did not believe that Mendel engaged in fraud and rejected Fischer's views. It's quite interesting to read, his research into that, took him 25 years, was published in a series of 6 papers, which are very interesting to read. So that was my first sort of encounter, or serious encounter with Mendel. Okay, so far to this little anecdote. Now, I think my talk is about protein degradation and the molecular machinery of protein degradation. Now, why is that important? I think for the healthy life of a cell, it is important that there's a balance between protein synthesis, protein folding, and protein degradation to maintain, and to respond, in its proteome to, environmental stress, or, to go through healthy aging, or, to a healthy development of organisms. And there are, of course, a couple of key elements, in proteostasis or protein homeostasis. I think I should have mentioned here also the ribosome. Then come the chaperones, which, are important for cellular protein folding. And then there are essentially 2 protein degradation systems. One is the ubiquitin proteasome system, and the other one is the lysosome autophagy system. So my talk will deal almost exclusively with this system here, which is important for the removal of misfolded proteins or otherwise damaged proteins, And I think equally important, at least, I mean, many of these proteins are, of course, prone to aggregation. And so, this is one aspect, but I think the other one, and equally important, is the fact that, many proteins, as the cell goes through its life cycle, are no longer needed and must be irreversibly removed. And so it terminates the lifespan of many cellular proteins. Now, very briefly, I think many of you know about the ubiquitin proteasome system. So ubiquitin, as you know, is a small protein which acts as a kind of a death signal via a relatively complex cascade of events, starting with the activation of ubiquitin, its conjugation, and ligation. Ligation is the most important aspect. There are several hundred genes, that sort of confers specificity in the marking of substrates for degradation. And then finally, we have to extend them because several ubiquitin molecules are required to act as a degradation signal. Now, these proteins marked with polyubiquitin then become, the targets of the 26S proteasome, which is the molecular machinery that is basically the principal machinery in the cell for the intracellular protein breakdown. Now, the next slide is a very simplistic view of the pathway. It's simplistic in the sense that it shows only what I like to call the executive arm of the system here. So we have the substrate, and that is recognized then by the 26S proteasome, which is a molecular machine of 2.5 megadaltons, so roughly the molecular mass also of a ribosome here. And in a process which I will describe in some detail in the course of my talk, in an ATP-dependent manner, somewhat surprising that protein degradation requires energy, but it does because of some of these steps here, which prepare substrates for degradation. It generates peptides, which are typically 8 to 12 residues long. Now, these peptides, are of very little use for the cell. There's one notable exception, that is the immune system. So for MHC class 1 presentation, some of them are very useful. However, even for being used by the immune system, they usually need a bit of trimming. They are usually a little bit too long. So there's another molecular machine which acts downstream of the proteasome, and that is tripeptidyl peptidase, or TPP2. It's even larger, it's 6 megadalton, but it's in its composition and in its basic structure quite a bit simpler. So the structure determination of TPP2 was quite a bit less challenging than the structure determination of the 26S proteasome. Now, it cleaves off tripeptides which have to be cleaved further. There's a whole array of aminopeptidases which sort of complete the catabolic cycle. So what I will do in my talk today is I will first talk a little bit about TPP2 because I will use it to sort of introduce the basic approach, which is a hybrid or integrative approach in structure determination, and then I will elaborate on 26S. First, studies of the isolated system, ex situ, if you like, and then, I will talk about structural studies of the 26S proteasome in situ in the context of the intact cell by electron tomography. Now, TPP2, I think I can be very brief on that. It was discovered something like, 30 years ago. It was classified rightly based on enzyme studies with model substrates here as a serine protease of the subtilisin type. It was clear that it formed very large particles at the time, but the structure was remained very poorly described, as a matter of fact. I think interest rose suddenly when, about 16 years ago, it was discovered that, if you inhibited the proteasome, then, TPP2, which is otherwise a relatively rare molecule in the cell, became massively upregulated. Now, so I think it raised the question whether or not, and this is still controversial, but probably a little bit misleading, that, TPP2 could at least partially substitute for functions of the proteasome. I think it's undisputed, even though, again, there's some controversy in the field, that it is involved critically in antigen processing, as I mentioned before. Now, I think very briefly, I think for those not so close to cryo-electron microscopy, I will go through the basic structure determination. So this is a typical electron micrograph of TPP2 embedded in a thin layer, 50 or so nanometers of ice here. So that is what it looks like. I mean, single particle analysis relies on the assumption that essentially when this molecule is in a thin layer of ice, it can assume essentially random orientations. So, I mean, for single particle analysis, as you will see later on, it is important that it occurs in all possible orientations. So you generate, so-called class averages showing, molecules which have the same orientation. And those class averages then, you don't need to do a tilting experiment in single particle analysis, but they have to be sort of given a place, in a virtual tilt experiment. So, once you have a place, and if you have truly random orientation, then I think you have perfect coverage here, but that would be sufficient. Then you can, by any of the established methods, generate a 3D reconstruction. Now, that was an early reconstruction of TPP2 about, a bit more than 10 years ago. At that time, most structures determined by cryo-EM were at best sort of medium resolution, so this was something like 20-25 angstroms at the time. But it's good and useful to describe the basic organization of the molecules. So as I said before, TPP2 is built only from one type of polypeptide chain, which has a molecular mass of close to 150 kilodalton, varying a bit from species to species. And you see that the molecule in its quaternary structure has these two strands, twisted strands, which make contact only at the end of the molecule here. And so, since the total molecular mass is, these strands are segmented, and you can simply count that you have 10 of these segments per strand, so a total of 20, and if you divide the 6 megadalton by 20, it's pretty clear that each of these segments must represent a dimer. That is the basic architecture, and then, of course, we try to solve the structure. We spent quite a bit of time trying to crystallize TPP2. What happens there, but this is a technical detail, if you like, this quaternary structure, as I just described, it exists in a relatively narrow concentration window. As you increase concentration, as you have to do in a crystallization experiment, then the strands begin to grow and you generate quite a degree of polymorphism, which is not conducive to crystallization. As you lower concentration, the thing falls apart. And you can do that in a very controlled manner so that you get dimers or tetramers. And then, of course, we wanted to use them as a starting point for crystallization, but then we had to modify them somehow such that they could not reassemble. Now, we did that very simply by adding octoglucoside as a detergent, and that allowed then to determine the structure of the dimer and the tetramer. Then we used, as shown here, basically the medium resolution EM map, which was at something like 8-9 angstroms at the time, to dock into it the crystal structure here of the dimer. Now, to describe the dimer very briefly, it is in yellow here. This is the N-terminal domain, which contains the active site, shown in red here, indeed, of the subtilisin type. There was an insertion forming, a little extra domain here, then a large central domain forming, the bulk of these strands here, and then a C-terminal domain, which was essentially in the monomer, a 12-helix bundle, so a 24-helix bundle here in the dimer. It's located, at the outside, and it's basically the number here of helices that determines the curvature. This is Drosophila, this is human. Human misses one helix, and therefore there's a little bit more curvature, and therefore, it's 9 dimers in a strand instead of 10. So that is the basic architecture of TPP2. But I think what I really want to explain here, and that is a hallmark of basically all intracellular proteases, is the following. The active site, in the intact structure, and it's best shown here in this schematic here, is deep inside those strands here. It's in what we call the catalytic cavity here, which is, so it's, well, sequestered from the environment. So any substrate to be degraded has to enter left or right here of these helical bundles here. It will, I mean, get to what we call the antechamber, and there are constrictions which are something like 10 angstroms in diameter, and that excludes basically any folded protein, so only, I mean, small peptides can enter and pass these constrictions here and make their way to the catalytic cavity. So, I mean, it is a size exclusion mechanism that is the basic sort of regulatory mechanism of TPP2. Now, an interesting thing or an interesting detail is that the dimer as such, I mean, as they also exist, of course, in the cell, is proteolytically inactive, and that is because, I mean, there is a loop here, the L2 loop, which, I mean, occupies the active site cleft here. So the dimer is inactive, and only as the whole thing assembles, and I mean, as the dimers get together into the strand, this loop is sort of pushed out of this cleft here, and then, I mean, the active site becomes active. So, I mean, it's a very neat mechanism in that activation always follows assembly and sequestration, which protects, I mean, from the cell from unwanted degradation. Now, that is a principle that also applies to the 26S proteasome, which I will discuss now in the remainder of my talk. Now, very briefly, that is a low-resolution structure of the 26S proteasome, I mean, which we produced something like 20 years ago, something again like 20 angstroms, not very revealing in terms of mechanistic aspects. So it comprises two subcomplexes. One is the 20S core complex or 20S proteasome. That is where the proteolysis takes place. And again, as you will see in a minute, there is a cavity formed here by the beta subunits, and that is, again, the catalytic cavity. So any protein to be degraded must somehow wind its way into that central cavity here where the active sites are located. Now, the 20S proteasome is in a eukaryotic cell not functional. It's a latent molecule, if you like. It needs to team up with, I mean, regulatory particles, one or two of them, as you will see later on. So, I mean, the canonical picture is with two, but that is a bit misleading. So their role is to prepare substrates for degradation, which includes the recognition of ubiquitinated substrates, their deubiquitination, their unfolding, and their translocation into the core complex. So that is the role of the regulatory particle here. So that is, well, the picture that existed until, well, relatively recently. So it even made it to United S stamps, even though they never asked for copyright here. And yeah, so I got it one day, I mean, on a Christmas postcard. Now, for the core complex, we had a crystal structure for 20 years now. So we found, I mean, a homologue of it, of the eukaryotic one, in an archaeon. And that, I mean, we were able to crystallize at the time. And so we obtained, I mean, a high-resolution structure of that, or 3.4 angstrom at the time, of the 20S proteasome. Now, we're at the challenge today to do this structure. We would not do it by X-ray crystallography. We would do it by EM, single particle analysis. This is a map of 3.2 angstroms we generated 3 or 4 years ago just for fun. So, but this, while this was, I mean, 4 or 5 years of crystallization trials here, this was 1 day of data collection and 1 day of data processing. So, I mean, things have become relatively straightforward. I think the important finding at the time was the highly unusual active site of the proteasome, and the unusual, I mean, it's a single residue active site. It's the N-terminal threonine that forms here the active site. Now, I mean, that was important and is still important because, I mean, knowing that it was the N-terminus, the very N-terminus that formed the active site, allowed very easily to design inhibitors. So invariably, all the inhibitors of the 20S proteasome, I mean, the principle is simply to extend the N-terminus by 2 or 3 amino acids. there's different chemistry in, if you like, in the so-called warhead of these compounds here, but they all do the same thing. They extend the N-terminus, and so they kind of muzzle the active site here. One of them, I mean, is this one here. I mean, under the name of Velcade, it became, well, a blockbuster that is used for the treatment of multiple myeloma. That was quite unexpected at the time. Now, I mean, that is the 20S, but that is, as I said, long time ago. Its structure was determined. Now, the 26S proteasome was much harder, and it took, I think, a couple of hundreds of man-years have been used in many labs around the world to obtain crystals of the 26S without any success. So we used at the time, I mean, 3 or 4 years ago, a hybrid approach or an integrative approach in which, I mean, the most important element was, of course, the cryo-EM density map, but it was only at medium resolution. It was only at 6 angstrom resolution. So it was not good enough to come up with an atomic model. So we used a hybrid approach in which another element was cross-linking and mass spec, and I will explain briefly how we used it to determine the structure. Now, well, why has it taken so long? I mean, first of all, it is the sheer complexity. It's, well, 2.5 megadalton, but I mean, it's, well, 33 canonical subunits here. I mean, very briefly, these are the subunits of the 20S. We ignore, for today, I mean, the many, in the meantime, there are roughly 10, 12 proteasome interacting proteins, some of them shuttling ubiquitin receptors, deubiquitinases, but I mean, we will talk only about the canonical subunits. Then, I mean, a very important element are the AAA ATPases. This is why the whole thing is ATP dependent. There are 6 different ones. They have non-redundant functions. Some are important for gate opening, some are important for unfolding the substrate. Yeah, so I mean, that is a module that basically drives the enormous conformational changes, which you will see in a movie in a minute, of the regulatory particle. Then there are 2 resident ubiquitin receptors. I mean, RPN10 and RPN13. RPN13 does not occur in all species, a little peptide that serves as a glue and an assembly factor. Then there is one deubiquitinase, RPN11 here. Then there's a whole module of so-called PCI module containing subunits. Their role is basically a structural role. They enable large collective movements of parts of the regulatory particle. And then there are 2 subunits, RPN1 and RPN2, the largest one, roughly 100 kilodalton, highly dynamic, as you will see, and their role is basically to serve as hubs for many of these proteasome interacting proteins. The real problem is not the complexity, the real problem is the heterogeneity of 26S preparations. And there are 2 elements of heterogeneity, if you like. One element is, I mean, compositional heterogeneity because of these substoichiometric amounts of all these proteasome interacting factors. But I think the more serious one is, I mean dynamics. I mean, it is the fact that we have very large conformational changes. So this is a low resolution map, and in orange you see what we call a variance map. So it highlights where the variations in the data set are. And you see they are invariably in the regulatory part of the molecule. You see the core particle, there's hardly any dynamics, and that is why it could be crystallized. Now, the first structure determination, I mean, that went in the end to a pseudo-atomic map of the molecule was, I mean, based on a 6 angstrom structure of the yeast 26S proteasome. I mean, a global figure for resolution is not very meaningful. In the blue part, resolution is better. In the red part, I mean, those are the dynamic parts. You see that is similar to the variance map. I mean, resolution was quite a bit lower. So we did some comprehensive modeling in which, I mean, these were the ingredients of the modeling. I mean, the electron density map here. Then, I mean, well, as I mentioned before, cross-linking and mass spec provided a set of distance restraints which helped, I mean, to interpret the electron density map. We could not obtain crosslinks for all subunits here, so we also generated deletion mutants, especially of the ubiquitin receptors, and then could generate difference maps to the wild type. And we did also quite a bit of crystallography of recombinant subunits where we needed a higher resolution, as you will see in a minute. So basically, the modeling went the following way. We had the electron density map, and we had a coarse-grained representation of all the subunits represented here as spheres, or as strings of spheres in the case of multi-domain subunits. Now, then, I mean, these subunits were sort of jiggled around within the boundaries of the map, and each and every configuration was then, I mean, scored against the violation of restraints that came from cross-linking. And then, I mean, we had some approximate position of the subunits, which were then refined by flexible fitting based on molecular dynamics. So we evaluated something like 1.6 million different configurations, and we came to an essentially unique solution here. So I think there were only 2 or 3 possibilities that did not violate the experimental restraints. And these 2, I mean, there was an uncertainty between 2 subunits. Now, as I said, in some cases, resolution was just not good enough to make any useful interpretation. So, for instance, the deubiquitination module, RPN11, was relatively poorly resolved, and I mean, it was the only subunits here that had, I mean, substantial amount of beta sheet, and this could not be interpreted, of course, on a 6-angstrom map in any way. The interesting thing is, again, and this is again the same principle, that, I mean, the active site here of this deubiquitinase here is in the recombinant protein, I mean, inaccessible. I mean, there's this insertion which forms a loop here and blocks, I mean, accessibility. So we have an auto-inhibition which is released only upon incorporation into the entire complex. Now, I think I will summarize this here by a little movie in which we first, I mean, disassemble the whole thing, and then, I mean, we rebuild it sort of brick by brick. It's not meant to illustrate in any way the assembly pathway. That is much more complex and involves a number of molecular chaperones, assembly chaperones. So this is, I mean, the alpha subunits of the core particle. In come now the AAA ATPases. Some of them have C-terminal tails here, which insert into a pocket here. And this insertion is what triggers the opening of a gate formed by the alpha ring, and that controls access to the interior, to the catalytic cavity. These are the 2 major hubs, RPN1 and 2. Now comes in RPN11 and RPN8. This is the deubiquitination module. They have, I mean, a large coil bundle here that associates, I mean, with the C-termini of the PCI-containing subunits, which form this kind of horseshoe configuration here and sort of embed in it the deubiquitination module here. And you will see later on why this is functionally important. And finally come the 2 ubiquitin receptors, which are sitting at the distal ends of the complex here. So they were probably recruited relatively late in the evolution of the complex. Now, that was the map, well, let's say, 3-4 years ago. Now, as you know, I mean, there have been a couple of technical advances in electron microscopy that led, as some people call it, to a resolution revolution. I think I would mention 3 basic elements. The first thing is, I mean, more or less full automation, enabling, I mean, to collect data day and night, I mean, in an unsupervised manner. The other one is, I mean, the direct detectors, which are, I mean, have much better detective quantum efficiency than, I mean, the CCD cameras. And finally, and I think that is important for my lab here, faceplates, which, I mean, improve contrast quite a bit. So I will show you basically, well, let me mention very briefly the Volta faceplate. That is something that was, I mean, developed by Radostin Danev in the lab. It's an equivalent of the Cernica faceplate as used, and many of you will know, in light microscopy. The problem is that, I mean, if you take in electron microscopy an image in focus of an ice-embedded sample, you basically have no contrast. So, I mean, contrast is generated, I mean, or was all the time generated by going into an underfocus. Now that leads to an undesirable contrast transfer function in which, I mean, some spatial frequencies are enhanced and others, I mean, there are gaps, there are zeros in the contrast transfer function, so it leads or generates a kind of a ghostly image. So, I mean, that is the kind of image we would like to have. And that can be achieved by using, I mean, a phase-shifting device in the back focal plane here of the microscope. So, I mean, the idea has been around for, I think, almost 50 years to create something similar to the Cernicke faceplate for EM, but I mean, all the designs in the past were completely unpractical. I mean, they changed their properties very quickly. Now, I think this is now the real situation. You see now the 20S proteasome here in a conventional defocus image, and this is a faceplate image. In this particular case, I mean, going to a very small underfocus here. So again, so it allowed, in the meantime, allowed to push resolution very easily to 2.2 angstrom resolution. And again, this was very fast. So you see, basically, it's 2 angstroms over most of the molecule, a little bit lower here at the periphery of the alpha rings. Now, so in the meantime then, I mean, we went to the human proteasome and using some of these technologies described here, and you see now we have directly obtained a 3.9 angstrom map. It's very hard to go beyond that now. I mean, the color coding shows the different subunits here, so we have now a map that can more or less directly be interpreted. So, I mean, you will see as we move on, this is the model based on the 3.9 angstrom map, and this is now the structure which is, I mean, more or less directly an atomic resolution model of the 26S proteasome. Now, we went on and generated, I mean, an enormously large data set. Oh, well, okay, there's one thing I forgot to mention, and that is that, well, maybe we could, well, I think it takes too long. There's one subunit, and that was this subunit here, RPN1, which was very poorly resolved. And I mean, this is one, again, one of these hubs, I mean, designed to interact with proteasome interacting factors. Now, what we can do with the faceplate is we can work with rather small molecules, and RPN1 is unfortunately, I mean, relatively, well, it's 100 kilodalton roughly, but we could, using the faceplate, at least come to a 4.3 angstrom map. I mean, the molecule is not particularly well behaved, I should say. I mean, human hemoglobin is much better behaved, so this is in the sense that it shows all orientations in the eyes, and so it was very easy with hemoglobin being a relatively small molecules by the standard of cryo-EM people to get to a 3.4 angstrom map. So, but I mean, we can fill some of the gaps now by single particle analysis of the components. Now, we collected a very large data set in order to look into coexisting conformations. And I mean, so we go through 3 phases, S1, S2, S3. S1 is, to cut a long story short, I mean, basically what we call the ground state of the molecule. This is, I mean, when the molecule is sitting there in the cell and waiting for substrate. S3 is a state in which it's dealing with substrate. It's engaged in processing substrate. You have to add either substrate or, I mean, you have to mimic substrate somehow. There's an intermediate state here, and I mean, you get already an impression of the very large conformational changes. You see, we have here a rotation of all the PCI subunits, and in the middle, I mean, the ubiquitinase here. And we have also a shift, which is important because, I mean, this shift sort of aligns the unfolding channel in the ATPase with the gate of the alpha complex, as I will show in a minute. So it performs, I mean, very characteristic movements driven by the ATP cycle here. ATP is the part that was in blue here. So the purpose of all these conformational changes is to bring the substrate, which is originally, I mean, because the receptors are sitting here at the distal end of the molecule, it has to be brought to the mouth of the AAA ATPase, because I mean it has to be channeled through the AAA ATPase to become unfolded. And oops, I think I'm losing my microphone. Okay. And then also the deubiquitinase has to be brought into close proximity of the mouth of the AAA ATPase, because I mean the ubiquitin has to be removed in order to go through this unfolding channel here. So I mean there's emerging now a picture, and I mean at least some rough understanding of, I mean, the functional cycle of the 26S proteasome. This is what I mentioned before. I mean, in one case here, the unfolding channel in the AAA ATPase is aligned here with the gate in the alpha ring. In the other one, I mean, it's sort of dead-ended, and under those conditions, substrate cannot be degraded. I mean, it becomes stalled here. Now, I think at this point I want to move to some in situ studies, and I think so the main interest of my lab over the past 25 years has been the development, as many of you know, of cryo-electron tomography. So we would like to be able to study macromolecules in their functional context, and that is, after all, the intact cell. So, I mean, one example here is we grow hippocampal neurons directly on an EM grid. So this is the EM grid, and these are, I mean, the processes of hippocampal neurons. Now, then we take a tomogram, I mean, and cryotomography, I mean, as many of you know, combines the best possible structural preservation with the power of relatively high resolution three-dimensional imaging. So this is a synapse here. Now, we can move into the cytosol here of this hippocampal neuron, and I mean you see now in the tomogram, this is without any post-processing, okay? This is the raw data. It's this molecule here enlarged a little bit further. These are slices through a 3D reconstruction to the tomogram of this molecule here, and there's another guy here. So you see that the quality, so we can obtain images now of the 26S proteasome deep in the cytosol of a hippocampal neuron. Now, I think what we would like to do with all that is to describe, the molecular sociology of the cell, as we call it. We want to describe and analyze the functional molecular neighborhoods in the cell. So for that, we have to determine the spatial coordinates of our molecules of interest, and we do that by systematic computational search of a cellular volume. I will illustrate, the whole approach of template matching with a movie, which is a bit old, something like 10 years ago, and so it illustrates the principle of template matching and the interpretation of tomograms. So it's illustrated here with data which come from what we call a phantom cell, which is nothing more but a lipid vesicle. And in this vesicle, we have encapsulated now a couple of molecules, proteasomes and GROEL and another protease called tricon protease. So we have a template structure, which can be a high or medium resolution structure of a molecule of interest, and we perform a systematic search by going through the tomogram, and for each and every particle we detect, we, by cross-correlation, determine the similarity with that. So we have a measure of the similarity. We can do it for one molecule and a couple of different molecules. So, that is the basic idea and the principle of, studying, let's say, the molecular sociology of cells here. I think I should probably cut that a little bit short now. So, in the end, so we obtain, a kind of a molecular map of the cell. Now, we do that for 26S proteasomes, in this case here, and of course they occur in all possible orientations in the cell. So these are the raw data of several proteasomes in different orientations. We can again generate class averages, and we can then obtain 3D reconstructions, and we can do classifications. If we do that in the hippocampal neuron here, this is a global average over all molecules. You see already that one regulatory complex is weaker in contrast than the other one. So the main difference in the variance map is the presence or absence of one regulatory particle, but there are also distinct differences in the other one related to their conformational state. So we can do a classification of the data set, and interestingly, in the hippocampal neuron, only roughly 1/4 of the 26S proteasomes, has two regulatory particles. Well, the majority, 75–6%, is what we call single-capped here. We can do the classification quite a bit further, so dealing now with, these differences in the other one, and then, we obtain two different conformations here, which we can correlate very easily now with the high-resolution structure we have from the ex vivo studies, and we can from that say that these molecules are in the ground state waiting for substrate, and these ones are, dealing with substrate. We can actually visualize and map where the substrate is located, but I cut that short. This is what we call a molecular census. So under the conditions of that experiment, in the absence of proteotoxic stress, the majority, is in the ground state. And also of the double-capped one, an interesting finding is it never happens that both ends are really dealing with substrate. It's only, one regulatory particle. Now, the nice thing is that we keep the coordinates of each and every molecule within the cell, and we can map them back to the cell, okay? So we can say, well, this is a mitochondrion. We can say, okay, here's a proteasome, it's in the ground state. Here's a proteasome that is active in dealing with substrate. Now, so we want to map functional neighborhoods. So we can go back to TPP2 now, and you might just see in the raw data you can already recognize, because it's in the right orientation, a TPP2 molecule. Here it would be difficult because it's, in a different orientation. But we can then generate an average without, any other information and input here. Now, TPP2 is much more challenging than the proteasome. It's because, the molar ratio of, which we can measure here, of proteasomes to TPP2 is roughly 15:1 or 20, in some cases 20:1. It's only in neurons that they are relatively abundant. And so we can generate an average. it's not yet high resolution, but it's about as good as, the single particle studies where 7–8 years ago, so it's of the order of 20 to 25 angstrom now. And again, so we can then do quite a bit of statistics and see, thus, the position and location in the cell of TPP2 and the proteasome, do they co-localize? And they do. This would be a radius here of roughly, I think, 300 nanometers. So we have what we call degradation centers in the cell, which are not membrane bounded, but there must be phase separations and other principles. Now, hippocampal neurons were easy in the sense that we could look at them in toto because they were thin enough. Normally cells, eukaryotic cells, are far too thick to be studied in toto. So we developed technology over the years based on focused ion beam milling, so we grow all sorts of cells directly on an EM grid. Then, we can observe what we are doing in SEM. We use, a gallium beam, focused ion beam to ablate material. It becomes a very complex workflow, going from freezing, vitrification to, identifying, the areas or the volumes of interest by correlative fluorescence microscopy. So to identify and, locate the areas of interest, then we can target these areas very precisely now by cryo-focused ion beam and cut a kind of a window into these areas, and then we can transfer them to the EM. This is still a fairly complex workflow and needs to be, it's not in this form yet commercially available. So what we do very often is we either cut or ablate sort of ramps into the cell, which are transparent, or we cut what we call lamella here, and so which are then, let's say, 200–300 nanometers thick. So they are kind of windows into the cell. So this is a typical lamella now in the transmission electron microscope of an algae, Chlamydomonas reinardi, which has the advantage that, I mean, the topology of organelles is relatively well defined for this species. So, I mean, then we can zoom into any region now of this tomogram here. We can zoom in here on a Golgi apparatus, for instance. So this would be the nuclear envelope. This is a nuclear pore. You see the outer membrane of the nuclear envelope is studded with ribosomes. Here we have ribosomes sitting on the endoplasmic reticulum. You see all the COP vesicles, I mean, which leave the Golgi here. What we can do here is now we can take what we call subtomogram averages. So we cut out. Ribosomes are always very easy to do. I mean, they have high contrast, they are abundant. And for instance, here we can easily collect, let's say, a few thousand tomograms of ribosomes, and we can do averaging. And you see that with subtomogram averaging, we can now go to resolutions well below 1 nanometer. So that is what we call structural biology in situ. Now, coming back to the proteasome, so we can study in this system, I mean, the cellular distribution of proteasomes. There are two, let's say, kind of hotspots where they accumulate. That is always in the vicinity of the ER. And somewhat surprisingly, I mean, in the nucleus. And in the nucleus, it's very specifically in close vicinity to the inner membrane of the nuclear envelope. So we have the positions there, and then we can do, again, template matching and averaging. That is a typical cluster at the ER, a degradation center. It's, I think, the equivalent of the ERAD system here. So, I mean, in this color here, this is the 26S proteasomes, the ribosomes. I mean, we are here less interested in gray. CDC48 is a disassembly factor, which is important in interacting with the proteasomes here at these sites. Now, in the nucleus, I mean, yes, we can again. I think, well, here we cut into the proteasomes. Here, you see, I mean, the majority is very close to the nuclear envelope. We can, and not only close to the nuclear envelope. I mean, they are very close to the nuclear pores. And so we can do averaging again. Okay, so I mean, that is an average of nuclear proteasomes. We have, again, the two different states of assembly, one and two regulatory particles. We have, I mean, the two dominant conformations. I mean, they are sort of highlighted or emphasized by, I mean, the position of RPN6 here, which is a clamp holding the regulatory particle and the core particle together. And many of them are directly physically linked to the nuclear envelope here, but that is very preliminary data. We are analyzing a larger data set. So, I mean, they interact very specifically via, I mean, this additional mass here with the nuclear envelope. And, well, when we map them back now to the nuclear envelope, you see, I mean, they're always surrounding sort of nuclear pore complexes. I think the role is essentially that this is the site where the inner nuclear membrane and the outer nuclear membrane are sort of continuous, but they must, I mean, have a different proteome. And so the diversity must be maintained. I think it's maintained by the degradation of those which should not go to the inner nuclear envelope. So, I mean, that is where we currently stand in what we like to call visual proteomics. And this brings me to the end. So I think that is basically the group that works on degradation machinery. I mean, many of them have left in the meantime. Sho is at MIT, Elizabeth is at UCSD, but that is the group of people who over the years have worked on that system. And we appreciate collaboration with Andrei Charlie's lab and Keiji Tanaka and Rudi Ebersold's lab. And I thank you for your attention.
Thank you very much. Thank you very much for a fascinating lecture, an excellent demonstration how modern structural biology works. I'm sure there will be questions from the audience.
Don't be shy. Yes, Jožo?
I'm just wondering, in one of your complexes you revealed several details by cross-linking. How in such big protein protein, you can so specifically cross-link those
No,
different
it's not
domains?
specifically cross-linked. I mean, it's cross-linked wherever. I mean, we generate a cross-link, but then we analyze it. And I mean, so basically the procedure is then to digest the protein. I mean, to separate all the peptides, determine by mass spec their identity, and there are ways of enriching the peptides which are cross-linked, and so then we obtain a set. I mean, if we do a cross-linking experiment, typically, well, we use different cross-linkers, of course, for different distances. We obtain maybe 100 or so cross-links, of which, I mean, some of them, I mean, we have to do some curation of the data sets. I mean, there will always be some false positives, but there are ways of identifying them and rejecting them. So in the end, I mean, with a system like the 26S, we use between 50 and 70 cross-links identified in that way, I mean, to sort of interpret the electron density map. So the cross-linking is not specific at all. But I mean, the peptides that are cross-link can be identified very clearly. I mean, it was a tedious procedure, and it's still being used. I mean, not only with the proteasome. I mean, people working on polymerase and others have done that as well in the meantime. I think it was the first example which we did with Rudi Ebersold and André Charlie of that kind. Well, we followed up and do similar things also in vivo, but I mean, given the fact that now resolution is so much better and we get right away into the 3 angstrom range, I mean, these kind of integrative approaches, I mean, are no longer really needed. I mean, the latest map on the human 26S we could interpret without, and I should say, by the way, it was of course facilitated by the fact that the density or the map of the human one and the yeast one were basically identical. The differences were minimal.
And from biological point of view, just curiosity. What happens with all those ubiquitins? Are they also degraded, or they just?
No, I think they are recycled. I mean, they have to be. They are cleaved into monomers, and then yeah.
Yes.
I mean, I'm also thrilled with how it's dependent on the energy. So, you know.
Yeah, I think the energy comes in, I think, not for the cleavage, it comes in for, I mean, the action of the AAA ATPases. It's basically the main ATP-dependent step is, of course, the unfolding of the substrate. So this is very similar to chaperones. I mean,
and is it evolutionary, so the energy remains roughly the same?
Yeah, that is a good question. I mean, first of all, I think, well, the 20S proteasome is, well, it exists, of course, in all eukaryotes. I mean, it exists in the vast majority of archaea. It is relatively rare in bacteria. I mean, there are only a few species in bacteria. Bacteria have many other ATP-dependent degradation systems, the CLIP system and many other systems. So I think, but there are a few bacteria which have, I mean, bona fide proteasomes. I mean, they also need, of course, an ATPase, but the interaction is more transient there. I mean, there is in archaea, at least again in most of them, there is a protein called, PAN, proteasome activating nucleotidase. So PAN is, I mean, a homolog, but a homohexamer, not a heterohexamer of the AAA ATPase that is in the 26S. So there are a few exceptions amongst archaea which do not have a PAN homolog, but then they have, I mean, they have other AAA ATPases. I mean, it was confusing for us because we had discovered the proteasome in thermoplasma in an archaeon, which, I mean, paved the way for solving a structure at the time. But then we could not find PAN in thermoplasma. So then we undertook the sequencing of the genome, which, I mean, 20 years ago was still quite an effort because we were desperately looking for this ATPase, but There was nothing in the genome. So there are 2 or 3 other molecules that can replace the function of the AAA ATPase, but they do not form any kind of a stable complex with the 20S proteasome. So they can interact only transiently. There's a molecule called VAT that does something similar. They all need an unfolding machinery, and that is the ATP-dependent step.
Yes, Karel.
The gate opening is less critical. I mean, it doesn't require.
With your method, can you give us actually absolute numbers about molecular complexes per cell? How, what's the dynamics of that? Whether it has any biological meaning? If you look at these, you can really now count the molecules per cell, right?
Yeah, we can count, but I mean, we have not done that yet because I mean, I think you have to keep in mind that I mean the volume we look at in a tomogram is small compared with the volume of a cell. Okay, so I mean, typically a tomogram is, let's say, well, it's less than a cubic micrometer, but the cell maybe is, let's say there's 2 orders of magnitude. I mean, now in Chlamydomonas, the situation is different. It would be a major challenge to, we can piece tomograms together of entire cells, but we mostly do it of relatively thin filamentous cells. But I think we will be able for Chlamydomonas to get a good estimate of the number of particles. I think I should not give a number at this point. I mean, we find, it's probably of the order of between 10 and 20,000 26S proteasomes in a Chlamydomonas cell. So the proteasome is an abundant protein. TPP2 is a rare protein. You saw the average was of only 90 particles, and for these 90 particles we had to take 70 tomograms. Okay, so typical we have 1 particle per tomogram. Okay, so that is where I mean, the quality of the primary data becomes very important because, I mean, our goal is that at the end of the day, and we are doing that testing with nuclear pore complexes, that, I mean, a tomogram of a single molecule is good enough to see enough detail so that we don't rely on averaging because many of these are highly dynamic, and if we average, I mean, we also lose some information, but we are able now, and I think we have published in a paper a few months ago, a pretty good nuclear pore structure based on only 4 molecules. Okay, so the primary data is getting better and better, and there's still room for improvement.
Yes, Lem?
So the ribosome or the proteasome facilitate you by remaining kind of standalone particles with the contrast. If they were incorporated, say, the proteasome into a body in a sick neuron or something, would you have any chance of being able to see it or follow it?
Sorry, could you repeat? I mean, I didn't get the first part of your question.
So these things, ribosomes and proteasomes, they remain standalone particles and you have the contrast. But if they were in some dense structure, like in a neuron, say the proteasome might get ALS neuron or something.
Yeah.
Would you be able to follow it?
Yeah. I think that is not a problem. I mean, currently resolution in tomography is always, dependent on let's say, the abundance of the molecule. I mean, as you see with the ribosome, you can relatively easily, collect a few thousand, and then you can go to sub-nanometer resolution. Proteasome, I think we can, we have examples there again in a neuron in which, we have overexpressed huntingtin, and then, proteasomes accumulate, and we have a fair number. There we get relatively easily to 12 angstrom resolution, and I think,
Yeah, in this context, maybe I have a question. Yeah. As you showed, it's a tremendous development of technology, how the resolution improved. And you showed with a faceplate, you can go down to molecular weight of about 100 kilodalton.
60.
16?
Hemoglobin was 60, and that was 3 angstrom, yeah.
Okay, 3 angstroms.
So
16
I think
kilo.
it's still not the limit, yeah.
Okay.
Yeah, I think, yeah, probably I think 40 kilodalton or so, then it becomes critical. But I mean, the improved contrast with the faceplate. For a long time, I think the feeling was that the limit for single particle analysis would be something like between 200, 300
Yes.
kilodalton. And then, I mean, 200 was easy without any symmetry. But I mean, with 60, we are very confident. I think probably, yeah, there will be a limit, but I mean, it will be 30, 40 kilodalton.
So it's a great future of the cryo-electron microscopy and also tomography. Any other question?
One question last.
Yeah, can you scream?
I would like to ask that this Volta faceplate is going to eliminate the use of phase contrast correction and all that, or it will automatically correct the phase contrast?
Sorry, again, could you repeat it? Or maybe it's better if you have a microphone. I mean, it's...
I can speak a little bit more.
Yeah, or you have to shout. So.
Thank you. Well, I don't know much about cryo, but I would like to ask this Volta faceplate, will it gonna help in the, like we have to phase contrast correction we have to do manually or something like that. So it will eliminate the use of that? Like by using Volta faceplate, can we eliminate this correction, or it will automatically do it? How it will work basically?
The faceplate?
Yes, yes.
Well, I mean, I think the faceplate, I mean, well, the basic principle is the same as with the Cernica one. I mean, you have a phase shift to generate phase contrast. I mean, for quite some time, I mean, I think the tricky thing in the use of the faceplate, but it can be fully automated, is precise focusing, okay? So you have to be truly in focus, or I mean, just a little bit of under focus. Now, that is more demanding in terms of automated data acquisition than, I mean, if you simply go to some kind of under focus and determine then the contrast transfer function and correct it accordingly. So I think the price you have to pay for that is basically that the data acquisition is about 2 times as slow as it is without the faceplate, because, I mean, you have to measure, I mean, the focus on a couple of spots surrounding, I mean, the area of interest, and that takes time. So, I mean, if you have a very large complex where you can very easily have enough particles, I mean, you have more pixels for the alignment and everything, I think you have to think carefully whether you want to do it with the faceplate or without the faceplate. But, I mean, as soon as you have either heterogeneity, a difficult specimen, or have small one, then I think it has dramatic advantages. Yeah. But you can automate it, if that is the question, as well, but I mean slower. Yeah.
Maybe another question?
Thank you very much for presentation. I want to ask, so like from technological point of view, like we wanted better resolution, there was invented the direct detector. We wanted better contrast without losing of quality, we invented faceplate. And or wanted to look into the cell, there was like FIBM. So what's the next checkpoint of microscopy from your point of view? Because you could see arises from the scratch.
I think first in single particle analysis, there's still room for improvement. I mean, we are not yet sure that we make the best use of the faceplate. I think there are still always tricks. You saw, I mean, that I think over the last year we went from 3.2 to 2 angstroms. With the direct detectors, I mean, there's also room for improvement. I mean, not big, but I mean, there's still, I mean, I think we might be able to gain something like a factor of 2 there, but I mean, would be already quite helpful. And these developments are underway. These will be available. I think in tomography, the challenges are much more on the sample preparation side. I mean, I think focused ion beam milling is working very well, and at least, I mean, for people experienced, it works reliably. But I mean, one deficit currently is that we are very good in targeting the right area in 2D, but not in 3D. Okay, so XY is easy, we can do it precisely, but I mean, we never know whether we cut it on the right height. So I mean, that is, we must have sort of, it must be guided by some sort of confocal imaging, but I mean, this can be done in principle. Yeah, I think another challenge is that we can currently do the focused ion beam only with cells which we can grow directly on an EM grid. So I mean, they are there, and then we can ablate material. The next challenge is, and I mean, it works in principle, but not to the extent that we can really produce data with that, is what we call the lift-out procedure. We would like to be able to have something like, let's say, a C. elegans embryo, and would like to be able to target any volume in such a multicellular organism, or let's say in a tissue, and find, let's say, and cut a window that accommodates, let's say, a centrosome or something like that. So lift-out procedures, I mean, and for anything larger than a single cell, which we can either grow on a grid or deposit on a grid, is still very challenging. So I think that requires quite a bit of development to find a volume in a tissue or something like that. So, but I mean, it's unfortunately there is relatively little support from the manufacturers. It all needs to be done in academia. By the way, that's also true for detectors.
Okay, I think it was a very nice and extensive discussion.